Very good to be with you today. So we humans have always had a very keen eye on recognizing traits in microbial processes that are of value to us. From fermenting yeast to medicinal plants, we've used the tools that are available to us to, yeah, I'm taller now. From fermenting yeast to medicinal plants, we've used the tools that are available to us to gradually refine our understanding of what it is that is the source of this value. We move from considering the source. For example, the bark of the willow tree was used to treat pain and fever and inflammation, to the understanding that it is a component of that bark, the salicylic acid that is the active ingredient. We synthesized a refined, less irritating version for human consumption, and we call it aspirin. In these searches, we have developed tools that translate living microbial information into raw data that we can process, refine, and search for value. My role is that of someone who has spent much time with data, with algorithms, understanding how they interact, and what we can meaningfully experience from their interaction. And when we say value, I think that we can generalize and say we're interested in the result. Most of us do not get to see the processes of science. The same with AI. We don't get to see in the proverbial black box. We just benefit from its working. However, I do believe there is value to be found in the process, and there is beauty in the processing too. And there is meaning to understanding what is happening inside the box. We should know what goes on inside the box, or at least have the opportunity. I feel that if we do not concern ourselves with these processes, if we distance ourselves or are restricted from the understanding of these processes, and therefore a true, deeper understanding of the world around us, are we humans as a whole really going forward, or are we going backwards? As an artist who has been entranced by technology, I want to help make these processes visible and audible and sensual, to open the box, even if just a crack, and show the workings. Not necessarily in a way of teaching, but an experiential encounter with the underlying mechanisms that we humans have meticulously developed. And I want to do it with honesty and with integrity, with respect to the data. And while data is just numbers, its meaning, its value is hidden and locked unless you look at it the right way. A work I made in 2016 is called "This Data Means Nothing." It explicitly plays on this idea that, and this data isn't random or fake, it was real data. It was sampled from an unconnected electrical terminal, laid out in a simple graph. Without context, our eyes and brain search for meaning. It's real data, it just doesn't mean anything. This piece plays upon our preconceptions and biases and reminds us that we must be honest with data and our representation of it, even as artists. So when we work with data like DNA, which is made up of just 4 bases, adenine, cytosine, guanine, thymine, we shorten it to the letters A, C, G, and T, and they're repeated millions of times in different combinations. We're presented with this imposing data set that is difficult to approach because of its size. This information can be extracted using the process known as whole genome sequencing and is delivered as a dense text file of A, C, and G characters. In this data is the instructions for how this organism will operate. Though it is not like a machine, it is a living, evolving instruction manual that has had revisions stretching back millennia. Many additions and deletions have been made, and there are sections that have become outdated but still carried along. And what can an artist do with such data? One approach is that we can translate it into a different form that is accessible and has meaning. From a data dump to a temporal experience, one that changes the terms of engagement for looking at the data as a whole, an overwhelming prospect, to one that is measurable in human time, in human terms. We can translate it into music. In our first exhibition of work around high altitude bioprospecting, which Anna mentioned, we invited Professor Nick Lohman, here's Nick, from the University of Brighton, and his team to join us. What did I say? I've even written it down. He's from the University of Brighton. Okay. His team's work with the portable MinION genome sequencer was used in the field during the Ebola outbreak in West Africa. And he was published, and with his team, in the journal Nature. He kindly agreed to demonstrate the device in public in the gallery as part of the exhibition. So we used an impinger, and this is a device for sucking in air and capturing particulate matter. So we sampled the air from the gallery, and Nick and his team then sequenced the captured DNA and delivered me one of these text files that was full of DNA fragments right in the gallery. And while they've been doing this, I was working on a live coding a DNA genome to music algorithm using my own node-based visual programming system called Fujio. They passed me the file, and I'll play the short sample of music generated from that DNA. Of course, this doesn't communicate any particular understanding of the instruction manual, but it does bring audience members into a confrontation with a manifestation of the DNA information that surrounds them. Like music, it is invisible and it's temporal. The algorithm was simple and naive. Adenine became the note A, cytosine C, guanine G, and thymine. There is no note T, so I use C sharp. And this type of arbitrary creative choice that many data visualization projects take reveals a point of weakness we must be mindful of. In my opinion, we should try to avoid assigning an arbitrary translation to data. But here, the notes are not the data, the music is not the art. The experience of the endlessness of it as one contemplates the genetic information passing around us and in and out of us through our breath is the artwork. And no one had ever done this kind of environmental sampling and performance before that I know of. So I expanded upon this idea with my installation here for the Francis Crick Institute in London, where I was awarded a commission to create a major interactive digital sculpture that was located in the front window of the building. The Institute is the UK Centre for Genomic Research and is named after Francis Crick, who was one of the discoverers of the double helix structure of DNA. On the front side of the sculpture, we can see here were 28 video screens that played a video piece that I had made from imagery from multiple labs, designed to encourage members of the public to interpret what they were seeing by hinting at natural environments such as fields of flowers, which were actually cells of fruit flies, and starry skies that were data plots from a completely different lab. So as the viewer physically moved closer to the sculpture, the screens would change, revealing independent videos of the inside of the building, the labs, and the hands of the scientists at work. And this last part, the hands, spoke to the hidden economies of labor in science, which are highly manual processes and procedures that are hidden from the public. And that I wanted to show and make explicit. This interaction, moving closer and revealing what was behind the science, was the core message of the work. The closer you look, the more information is revealed. On the back of this sculpture, well, that's another shot. This is on the back of the sculpture, was a DNA clock. I'd used the genome of a Pombe yeast, an organism that is used widely across science as a model organism. And I played out its genome across the back. It took a full year for the genome to play out, slowly shifting by with different colors assigned to the base pairs, shifting in hue depending on the hour of day, the day of the month, and the month of the year. As I learned more about DNA structure, I understood that there were other structures present in the DNA beyond the double helix. Codons are groups of 3 DNA or RNA nucleotides that specify an amino acid or a stop signal. And these gave me the idea of adding a new level of structure to my genome music. 3 notes form a triad, a chord. This idea of translating codons to chords seemed to be more meaningful, at least to me, than playing back single notes. It conveyed something about the actual structure of DNA that related directly to an aspect of our Western music theory. So for the Fermenting Futures project that Anna and I worked on together, I created a new sound piece based on the work by Dr. Ozg Atta of Boku University, who identified and extracted the fermentation mechanism from the Saccharomyces cerevisiae yeast, also known as brewer's yeast, the only fermenting yeast, and successfully transferred it to a different yeast, Pichia pastoris. I took the DNA that she had worked on and translated it into this music based on the codon structure, and then subtly accompanied the genome on guitar as an art-science duet. So anyway, to summarize, bioart isn't just wetware. The crossover with data processing algorithms, including AI, also present amazing opportunities to engage audiences in the revolutionary scientific advances that are being made. But the value is only understood with the right way of looking at them. And such art science projects can help give everyone new ways of looking and understanding. Thank you.